Hi, I'm Professor Siva Krishna Vanjari. I'm a faculty here in IIT Hyderabad. I work in the area of biosensors, and this is a combined work in collaboration with Professor Shiv and Singh. And he's actually not feeling well. So he said, why don't you present? I said, okay, fine. I'll do it. So, instead of running through, the next presentation is also from our group itself. So what I'll do is I'll just combine the gist of what we are trying to do and then try to give it as an example, right? So if you actually take any sensor platform per se, right? Let's actually kind of dissect the innovations that are happening in sensors field, right? There are a set of sensing technologies that are available, right? That we have, right? So it be it electrochemical, optical, and all those things, right? In electrochemical, people will say, if I have done DPP, why are you not doing EIS? Both are same, right? You have an extra graph that you are actually presenting out there, but both are same, right? You can extract the same similar information from both the results. And more often or not, uh, the innovation happens in the material perspective, right? So that the novel materials would actually result in enhanced sensing capabilities, selectivities, and the other aspects. And the other thing is, if you were to actually look at a sensing mechanism per se, right? What would be the ideal sensing mechanism? And that could actually help us in taking it to the next level, from paper stage to a product stage, from idea stage to a technology stage, right? I mean, that implies that you have to actually look at technologies that are compatible not only in terms of sensing aspects, but also in terms of the associated peripherals such as electronics, right? If the associated peripherals are huge and if your sensor is miniaturized, right, then the advantage would be minimal, right? Agreed that the sensor is miniaturized, but the associated peripheral is actually big, right? So, in that particular sense, the most compatible methods with electronics would be something like electrical characterizations or electrochemical characterizations, wherein miniaturization is possible immediately. So then comes, as I said, right? So you look at mechanisms that are the sensing transduction mechanisms that are close to miniaturization and combine that with novel material chemistry and novel materials. That would actually give a low cost platform, right? Can we actually build low cost platform? And then the next set of things that would actually come in is what is the application that you're actually targeting, right? Is the application, significant enough, right? That it would actually bring in a change in the quality of life. Right? These are the three questions that as a sensor researcher that we should actually ask ourselves. Materials, right? The peripherals and the application, right? So in this particular case, the question that we have actually asked ourselves is, what is the application to start, right? Criticality is very, very important, right? Time is money, right? So in accidents, more so, the time is money, right? So how how damaged is the brain, right? How dramatically the brain got injured, right? So the way to predict is there is an index, right? I don't recollect the name exactly, wherein you can actually give a scale, but all that needs is a doctor diagnostics followed by an MRI or a CT, right? So this is a valuable time. So instead of that, can we actually assess the level of injury using a biomarker, right? So the innovation happened at the biomarker level. There are two biomarkers that are being approved. One is UHC1 and the other one is GFAP. Now that the significance of the biomarker is understood, right? What needs to be done? As I said, then there are two aspects. This first faction is what is the technology that we would like to adapt, and what is the material that we would like to adapt. So, I mean, I've covered all these things by talking itself, right? So, the technology that we are actually using right now is a capacitance. Anywhere, right? If I were to look at the sensor platform per se, right, what you're looking at is at the interface, there is a reaction happening, right? There is a bioreceptor and there is an unlike molecule, they are interact, right? There is a tangible change because of the reaction between the bioreceptor and the molecule. Sometimes they actually bind each other, and then whatever is beneath is actually providing you that tangible change. So what is that tangible change, right? And if we were to actually increase the sensitivity of of your device, you have to focus on two things. The first thing is, how are we actually maximizing the interaction between the bioreceptor and the material itself, and the analyte. And after the interaction has happened, what is the mechanism that would actually eventually give me, if there is a delta change here, what is the tangible change that I'm able to measure? And how small would that delta be, right? So that's what we ought to look at. So when we look at, and then comes the materials aspect. When you look at materials aspect, you always try to go for a nanomaterials. The simple reason being surface area to volume ratio, right? For the same surface area, you have more and more reactant surface available. That means to say that you could actually immobilize antibodies or bioreceptors more, and then after that, you can actually do. Now, taking into the advantage the cost, right? So what is the right combination of doing this? So if I work with materials like conductive polymers like polypyrrole and others, right? And then add this additional factor that would actually increase the kinetics per se, you have a nice combination. Now, after that, what you ought to do then is to immobilize the antibody. You search everywhere. The whole literature, right? I'm a non-chemist, right? So I know about EDCNHs. I've been doing it for almost like a decade or so, right? EDCNHs chemistry will be there. Glutaraldehyde chemistry will be there. You can actually hand count the number of surface functionalization methodologies. One or the other, you have to choose, right? So if you have an COH bond, then you actually look at converting it into a CONH bond using EDC and NH chemistry. If I have an amine bond, you just use glutaraldehyde to get the antibody immobilized on the surface. And that's what is actually done and will be done till a new surface chemistry emerges out that would enable the immobilization of all these bioreceptors, which are nothing but protein or peptide molecules, where the focus is on either amine or carboxylic, right? Till the chemists tell us, look, there is a new methodology of linking these things, right? We are more or less stuck with this, correct? Now, what is the advantage that we need to actually showcase right now is the robustness and the cost-effective line, right? So this is actually simply done. If the platform is a PCB platform, it is compatible with the electronics. All the electronics are actually assembled in the PCB platform. If I can combine the PCB platform not only for the sensing, but also inherently electronics would actually come into picture directly, right? These two combinations will take us to the next level. So with this in mind, so we proposed a concept of this capacitive detection. What do you mean by a capacitive detection? You have two electrodes, right? The dielectric property between the two electrodes have to change. whether it is a vertical capacitor, right, metal oxide and a metal capacitor, or a interdigitated electrode, metal, some material dielectric in between and a metal, or a non-conductive stuff, whatever it is, you are actually looking at a change in capacitance as a function of frequency. And how should the change in frequency happen? Because of the immobilization, right? Herein, if you look at it, the structure per se, you can actually see the under the layer view. So these are actually interdigitated electrodes, where you can take two set of electrodes and then measure the capacitance in between, right? So in some sense, this is a multi-analyte platform. You can actually put a drop here. Functionalize with a different material and a different antibody and do the same capacitor sensing. Correct? So this becomes a unique platform which also enables a multi-analyte detection. And how are we actually measuring it? When I say change in capacitance, I am actually using a simple impedance measurement unit, right? That you have a simple impedance measurement that happens, right? And the rest of the graphs, right, would actually talk about how the impedance is actually getting changed with the agglomeration of the particles and what are their features. The important point that I would like to highlight here is that all these actual measurements are done in plasma. So that means to say that we have the optimizations are actually done. Typical papers would actually start with a saline and then look at the other aspects, right? PBS to start with, and then how would actually the system would behave in a real samples, right? I'm almost done. Good. Okay. So in that particular sense, you can actually see that over a wide range of detection, you can actually see a change in the capacitance per se, and that is calibrated. What is the initial capacitance versus the change in the capacitance in the real samples is looked at. Now, these are actually done over months together, right? To understand whether there is a stability, right? And essentially, what is the limit of detection? Into few femtograms per ml. That means to say that if I were to make it as a product and then do all the clinical tests, we are well within the range of taking it to the next level. I would actually stop at this particular point and then right, we'll go on to the next topic.